So, hi everybody. I'm actually Garrett Frampton. Roman's my boss, and he sent me in his stead. So that was a late change to the program. So, as was said, I'm here on behalf of Foundation Medicine. We're a cancer diagnostic company in Cambridge, Massachusetts, and I'm a scientist there. Just a little bit about my background. I grew up in Boston. I spent most of my time there. That's my front yard last year. I really hope this winter does not turn out like that, but we'll see. Just to give a brief agenda of the talk, I'm going to give a little introduction to molecular diagnostics and oncology, and then highlight generally some of the key aspects for doing this successfully, and then I'm going to go into detail of a couple of examples of more detail about these topics. So, the introduction. So, I think everybody here knows that oncology therapies, targeted therapies that go after the specific molecular alterations in a patient's disease can be remarkably effective. There's a bunch of examples up there. The BRAF V600E mutation, for example, can be targeted with a targeted therapy, and these are coming online more and more rapidly. There are dozens that are available right now, and over the next 10, 20 years, there are hundreds in development, and hopefully the majority of those can be brought into the clinic. This is part of a broad trend in oncology where the sort of traditional histological understanding of disease is being transformed into a combined histological and molecular understanding of the disease. So lung adenocarcinoma is probably the best example, where in the last 10 years we've moved from a relatively homogeneous disease to a disease that's understood to be driven by a large collection of different mutations, each of which is either currently targetable by a therapy or is under investigation. And this offers tremendous opportunity for the patients, but it also creates a lot of challenges diagnostically because you need to simultaneously assess the mutational status of all of these genes in each patient. So yeah, the first challenge diagnostically is that just that there are lots and lots of genes for which the mutational status needs to be assessed. In lung cancer, here's a relatively comprehensive list of the genes that you might want to know the mutational status of in order to decide on the appropriate treatment for a patient. And this challenge is only going to grow as more and more genes are known to be relevant and known to be targetable. There's another technical challenge with this type of diagnostic. Unlike assessing germline genomic alterations, which are present in 50% of the material that's assessed, tumor specimens are heterogeneous, they have normal tissue in them, so you need to be able to assess mutations that are present in only a small fraction of the DNA that's present in the specimen. Another real challenge with cancer diagnostics is that the tissue that's available can be very small. The way that it's prepared, formalin fixed and paraffin embedded, can degrade DNA and prove challenging for molecular diagnostics. So any sort of sample prep or diagnostic methodology has to work in this setting to be useful clinically. For research uses, you can pick and choose the samples that you use, but for a clinical diagnostic, you need to get a successful test on all the material that's available. So what Foundation was, company started 5 years ago to create a diagnostic test to meet these challenges. And I'll just go very briefly through the workflow. You start by extracting DNA from the generally available biopsy specimens, starting with as little as 50 nanograms of input DNA. Then you do sequencing library construction and hybridization-based target capture to pick just the regions of the genome that you're interested in assessing. Then these libraries are sequenced to high depth using the Illumina HiSeq platform right now. And there are mutation detection modules that are used to identify each of the different classes of alteration that are present in a tumor specimen. And then a really important part, the job isn't done when you identify the mutations that are present. The analysis, annotation, reporting of those mutations is critically important to give treating oncologists all the most important information right up front. So I added this section of the talk to just highlight not a fully comprehensive list of the key elements, but what I saw as some really important things that you all may or may not have thought of in doing this in a clinical setting. So A lot of aspects of the laboratory methods are important, but really critically important is obtaining high sequencing coverage across all of the test regions. So that means really getting even sequencing coverage. If you get extremely high average coverage, but 10% of the regions that you're looking at aren't covered at all, that's going to be a big blind spot in a diagnostic test. Another really important challenge is removing the PCR duplicates that are created during sequencing library construction. If you sequence the same input molecule again and again, the analysis, if you treat each of those as independent data points, the analysis is not going to work properly. So there are a couple of different methods that can be used to remove these PCR duplicates. If you used hybridization capture for getting your target regions, you can use the unique start and end points of the fragments to identify PCR duplicates. If you're going to use an amplicon-based approach, you can use a random fragment molecular barcode to identify PCR duplicates, but that's critically important. Another aspect of doing this properly is having appropriate variant detection methods. So that means detecting all of the relevant classes of alteration, being able to detect alterations that are present at very low allelic fraction. We use local assembly for indel detection. I think that's the optimal approach for identifying short insertions and deletions that are present in cancer genomes. Also, if you're going to be calling copy number alterations, you need to be able to distinguish focal copy number alterations. Those are amplifications that affect half a megabase, a few megabases, and those are typically the drivers of cancer, and that's different than polysomy in which a whole chromosome is amplified. The focal amplifications are much more generally treatable with targeted therapy. Quality control is very important. So at Foundation Medicine, all the samples that come out in the door go through pathology review. That's to assure that the specimen that we're testing looks like the tumor type that we expect it to be, and also to ensure that there's sufficient tumor content in the specimen to expect to have a good test result. We do gender concordance to make sure that the gender of the specimen matches what we expect the patient to be. Another thing we observe is that a small fraction of the samples that are submitted to our lab actually have multiple genomes in them, and that's because a small amount of material from one tumor, maybe on the microtome or elsewhere in the pathology lab, can contaminate. You can get cross-sample contamination, and if you're doing a diagnostic test that's sensitive down to 1 or 2% allelic fraction, that starts to become problematic. So if you have a sample that's 2% some other human genome, you can't call mutations in that specimen at 2%. So we've developed algorithms to identify this. If we can, we go back to the tissue block and retest, but if that's all that's available, you have to raise the threshold for calling alterations. I think another thing, doing this clinically, they're having a dedicated quality assurance group to follow up on any errors and improve processes as any sorts of errors or mistakes are found. Assay validation is critically important. I'm going to talk about that briefly. I think doing this in a peer-reviewed fashion with publicly available data is the only way that we as a community can trust each other's results. I think it's also very important that the validation material that's used is representative in mass and in quality of the material that's actually going to be used clinically. Also that a diverse set of alterations are tested, so you can't test a couple of alterations and then say your test has been validated across the full spectrum of genes and alteration types on the test. And also, this is a fine point, but specificity for a genomic test really needs to be reported as positive predictive value. If you're talking about the per base pair specificity of a test for a test that's covering 1 million bases, that's misrepresenting the truth, and so I think PPV is the appropriate way to talk about false positive rate in a genomic test. And then the other example that I'll talk about is the importance of proper variant annotation and reporting. So there are sort of well-known canonical mutations in cancer, such as indels in EGFR or fusions in the ALK gene. But for both of those types of mutations, there are non-canonical mutations which are similar to those well-known mutations but not exactly the same. So EGFR indels that are not exactly the same as what's typically seen are actually much more common than perhaps people appreciate. Or fusions of ALK to other genes other than its common fusion partners are also seen. So in order to really get appropriate therapies to as many patients as possible, you need to identify not just the really common canonical mutations, but all of the other possibilities. Germline variants typically aren't important, but there are a number of pathogenic germline variants, so you need to filter out the germline variants that are not important and report the ones that are pathogenic, so that's critically important. And then, as I said, this field is changing rapidly, so a test and a testing platform needs to be able to rapidly incorporate new knowledge, both in terms of driver mutations or connections to therapy or clinical trials or other knowledge. So a test needs to evolve as the field is evolving. So I'm going to give you a little example of analytic validation. I'm actually not going to talk about the specifics of the data much from our company that's been published, but I just want to talk to you a little bit about how we approach this problem. So a key challenge in validating a next generation based genomic test is how do you validate a test that covers 1 million bases of genomic space. It's a lot more complicated than validating a test that detects a single mutation. Tumor specimen can have a mutation of any of these types at any position in the test region and at any mutant allele frequency. So getting actual reference specimens that reflect this full diversity is not possible. Those material are not available currently. So we had to use a number of different novel approaches to validate our test. And like I said, I'm not going to go through the specifics of the data, but I'm going to talk about the 3 different types of experiments we did. So first is using pools of cell line DNA or using cell line DNA to model somatic mutations. So if you start with, this is just kind of a simple example, if you start with a pure cultured cell line that contains a genomic variant of interest on one of the chromosomes, so that's shown in red up there, if you tested this specimen, it would be present at 50% mutant allele frequency. But if you pool that specimen in equal part with DNA from other cell lines so that you dilute the mutation of interest, you can model somatic mutations at any mutant allele frequency. So in this example, the mutation on the chromosome shown in red would now be present at 10% mutant allele frequency. And so you can use this sort of approach to create specimens for validation purposes that go down to very low mutant allele frequencies. So for the validation study that we published, we used normal cell lines and used the germline variants that are present in them to model somatic base substitutions. And we used cancer cell lines with known indels in the genes that we're testing to model somatic indels. And again, we used cancer cell lines with known copy number alterations in this sort of pooling strategy to model copy number alterations and fusions. And so so this is using cell line DNA, and this was, I think, the meat of the validation study we did showed results from these sorts of experiments. But again, critically important is showing concordance to other testing platforms. So we did that, showing concordance to a number of different other clinically used testing platforms. And then finally, showing assay reproducibility. If you don't have good specificity, you will not get good reproducibility. So this is showing data from repeated testing of the same sample, but this sort of approach where you take a sample, maybe big, maybe small, and test it a number of times to ensure that the results are identical or assess how identical the results are another really good way to validate an assay. So these are the specifics of the Foundation Medicine test. Base substitutions are relatively easy to detect. We're detecting the vast majority of them at allele frequencies of 5% above. Indels are harder to detect. Copy number alterations are harder to detect still. So those are the specifications of the test, but I think critically important that the positive predictive value is very high. You need to be able to trust the results. If you see a mutation call in a report, you need to be able to trust that it's truly present in the specimen. So we published all this data a couple of years ago, and if you guys are interested in any more of these details, it's in this publication. And then finally, this is an example. I think it's exciting science. I think it also highlights the importance of proper reporting for the purpose of diagnostic testing in cancer. It's not simply enough to detect all the mutations, but you need to highlight the ones that are clinically relevant. It is really not. We need to partner with the treating oncologist to help them get all the information that they need and help them keep up to date with the most recent literature and knowledge. So just to start at the beginning of this story, the MET gene is a receptor tyrosine kinase that's fundamental in regulating cell growth and development. Point mutations in MET and copy number amplification are well-characterized drivers of cancer in a number of different tumor types, and these tumors tend to respond to anti-MET targeted therapy. So somatic mutations affecting the splice sites of exon 14 were first characterized in 2003 in lung cancer specimens. This is really a unique mechanism. Splice alterations typically cause loss of function, but here you have a gain of function caused by a splice site mutation. And what happens is, if you have a mutation in the splice site of exon 14, you skip that exon in RNA splicing. You get an in-frame deletion of a 47 amino acid portion of the protein, and that protein is required for degradation of MET. And so losing that portion of the protein, you have accumulation of the MET protein in the cell and hyperactivity of MET signaling. So like I said, these were first discovered in a research setting in 2003, but I think the clinical significance of these is just now being appreciated, and when these sorts of mutations are detected, it's critically important to highlight them so that the patients will receive the best targeted therapy. So we did a lot of work around MET exon 14 alterations, and I'm going to talk a little bit about that. So we performed comprehensive genomic profiling. This data was pulled together when we had profiled about 38,000 tumor specimens. This was up through February 2015. I think we just crossed 60,000 recently. So we looked at all of the alterations in all of these specimens and examined those that were likely to affect splicing of MET exon 14. And in total, we identified 224 alterations in 221 specimens. And I think what's really remarkable here is the incredible diversity of alterations. It's not like KRAS where you see the same mutation over and over again. There were 126 different sequence variants represented in these 221 patients. So detecting them and properly reporting them is critically important and very challenging. The alterations had base substitutions and indels at the splice acceptor sites and at the splice donor sites, and also non-coding alterations in about a 25 base pair region immediately adjacent to the splice acceptor site. And there were also 5 samples that had whole exon deletions of MET, so an indel that encompassed the entire exon. The indels were predominantly deletions, but there were also insertions and complex indels found in this set of data. So this is the distribution of these alterations among different cancer types. They were present in about 3% of lung adenocarcinomas and about 2% of other lung neoplasms. They were also present in about 0.5% of brain glioblastomas and 0.5% of cancers of unknown primary origin, as well as single MET exon 14 alterations identified in those other tumor types. So clearly, at least in lung and in brain, assessing the mutational status of MET exon 14 is critically important. This takes a second to load. It's a big figure. I guess this is big data. So this is looking at co-occurrence and mutual exclusivity among all the genomic alterations, including these MET exon 14 alterations that we found, and this is focusing on the cohort of 4,400 lung adenocarcinoma specimens that we had profiled. So this is how one of the ways we proved to ourselves that MET exon 14 alterations were clearly primary drivers in these specimens. What we saw, the figure on the left is showing co-occurrence and exclusivity. Basically all of the well-known drivers are mutually exclusive of each other, including MET exon 14 alterations, but also point mutations or MET exon 14 alterations were significantly enriched with copy number amplifications of each of those genes. And now the panel on the right is showing each of these driver alterations co-occurrence with a bunch of the other common mutations in lung adenocarcinoma. And what we saw here is that each of these drivers has a different pattern of co-occurring alteration. Strikingly, that red dot down there, about 45% of the specimens with MET exon 14 alterations harbored co-amplification of MDM2 and CDK4. I don't know what the importance of that is. I'm excited to find out, but I think that is further evidence that this is a distinct molecular subtype of lung cancer. So here's the most exciting part of the story. Earlier this year, there were 3 different case reports demonstrating clinical response to crizotinib in lung cancer patients with MET exon 14 alterations. So these were case reports at the beginning of the year. And then in the August issue of Cancer Discovery, we published the results that I presented here, including 3 cases of response to 2 different targeted therapy agents in patients with MET exon 14 alterations. And in the same issue of that journal, a group from Memorial Sloan Kettering published 4 additional cases of response to targeted therapy. If you're interested in more details about this story, there's the publication. But excitingly, there's now several different clinical trials underway to identify what are the best targeted therapies for these patients. Yeah, and so this is just the details of the patient cases that were highlighted here. So just to summarize the MET exon 14 story, these are important drivers of oncogenesis that occur in about 3% of lung cancers. They have remarkably diverse sequence composition, I think, really 120, everyone is different more or less. So appropriate diagnostic testing is necessary for detection in the clinical setting, but simply detecting the alterations is not enough. In order for all the patients to receive the appropriate targeted therapy, these alterations need to be highlighted. Just showing the genomic coordinates of the mutation is not going to be enough for doctors. So we need to highlight in the test reports, connecting this to the relevant therapies and medical literature. Yeah. So I think I showed you this figure at the beginning of the talk. I think what these results show is there's another slice of the pie that we can cut off of patients with MET exon 14 alterations that are excitingly responsive to targeted therapy. So, yeah, thanks a lot. You want to plug in? I'm staying here for questions. I'm just letting him plug in his computer.
Thank you very much. I do deeply apologize again for that mix-up. I read off the wrong sheet of paper.
No, it was on the. It was on the paper that you read. What could you do?
Yeah, as I said it, I noticed. I can promise I know the 3 other speakers, so that won't happen. Yeah.
Well, now you know me.
Question. May I start with one?
Sure.
Just going a bit further or a bit larger than the MET story. In general, if you typing these patients across the board, how many lesions do you actually find, and how many of those do you perceive currently as actionable?
So we divide our reports into 2 different sections. The known drivers of cancer, these are known recurrent alterations. They're typically 2 to 4 in the genes that we're testing, although that range is probably 0 to 20. Some cancers are very highly mutated, typically 2 to 4. And then there's a section of, at the back of the report, these are what we call variants of unknown significance. These are mutations that were detected in the tumor but that have not been observed before. There's nothing to say about them clinically, scientifically, and those typically are 4 to 10 per specimen. The test that we're doing is the entire coding region of 300 genes. So across the genome, there's going to be a lot more variants of unknown significance. I think the genes on our test cover everything that's currently known to be clinically relevant. So those sort of recurrent known mutations won't increase in number by increasing the size of the test. I think the real meat of your question is how many of those are clinically actionable. There are drugs for them. Maybe half of those alterations that we present on the front page are connected to targeted therapies, ideally in the disease that the patient has or in another disease. Clinical trials are a less excellent option, but a number of mutations can connect patients to clinical trials as well. So depends what you consider to be clinically actionable. So probably about half of those 2 to 4 that are presented on the front page of our reports.
Thank you. Great talk. So I'm from Salt Lake City, Utah. I had a question about, so how often do you find in your testing any indication of a germline mutation that reports then that there's a hereditary syndrome, such as Lynch or a BRCA mutation or something like that?
So,
or have you guys looked at that?
I don't know across the whole panel what the answer to that is. It's a large portion. It's not as much as a quarter, but it's probably more than 10%. But it varies a lot from tumor type to tumor type. In breast and ovarian cancers, BRCA mutations are quite common. In a lot of other tumor types, germline alterations are very uncommon. Some colorectal cancers, we see hereditary APC mutations. So.
And do you call those separately in your report? Do you make it very clear that there may be a germline alteration to encourage familial testing?
So we try to. We're struggling with what the right way to report that is. So we test just the tumor specimen. We do not make the determination whether a particular mutation is somatic or germline in that patient. And so on the report, we will say this mutation has been observed to be germline. We advise doing germline testing. I think the way we do it in the text section of our report is missed by some physicians sometimes. So we're trying to figure out what the right way to highlight that is without confusing people as well, because this is not, there's a lot of different levels of understanding of this material. And so figuring out the right way to report that to all of our customers is a challenge. So.
So I have a question. So I suppose not all the mutations at the genomic level are also expressed. So can you comment on this, and are you looking also for expression at the protein level? How it can be relevant for therapy?
Yeah, so our test does not, we sequence genomic DNA. We don't do RNA expression or protein expression-based testing. I think it's relatively safe to assume that the key drivers of cancer, when you identify them in a cancer specimen, they are being expressed. It would not make much sense to have a cancer with a KRAS mutation, codon 12 mutation, for that not being expressed, but it's true. That is another layer of complexity on all of this, that the more information, the better. So.
I think in the interest of time, we should probably move on. Thank you very much again.
Thank you.